Cancer's clever. It hijacked this mechanism and made it more favorable for them to grow and more aggressively and takes over. But now we have ability of understanding how this language and interaction happens, and if we can remove the hijacking and hijack it for our own benefit and disrupt their hijacking, we may be able to save people from dying from pancreatic cancer. And the exciting bit is this is a new way of thinking about treating cancer, and it is through new ways we can find new discovery that may be the Achilles heels to pancreatic cancer that we haven't found today. My name is Michael Lee. I'm one of the medical oncologists or clinician scientists working at the Peter MacCallum Cancer Centre in Melbourne, Australia. I spend my time looking after patients with upper GI cancers and neuroendocrine tumours, and my research interest is in pancreatic cancer, where I do translational research, doing the clinical trials in clinic and working in the lab to find out the mechanism behind the drugs. I think the first turning point is really as a young doctor, figuring out what does it mean to, what kind of doctor do I want to be. The first turning point is realization, what is medical oncology? I really actually stepped into medical doctoring because I want to be a psychiatrist, because it actually offers care and you really understand and get to know your patients. And that's what oncology offers, the art of oncology. That is really getting to know your patient and working out what is good for them. And what is good for you is different to what is good for me. And it's working out alongside them as their partner, and with the knowledge that I know, and with all the different fancy drugs I can do, I can help them in many different ways. And that's really what led me to do oncology. We have exciting research that's constantly changing. In my young professional career, the first standard lines of treatment of different cancer type constantly changes. And I joke and say, look, .com days were terrible for the computer scientists because they constantly have to throw out manuals. In oncology, that is probably the worst part of my job. It's now less than a year, I might have to change what I do from day to day for what is the right treatment for my patient, because information is just evolving so fast. But that's also the excitement. We really are making differences in oncology care. So that's the first landmark for me. That's really turning point. The second changing point is probably going to Vancouver to be at BC Cancer in Canada, where this is world leading center for a lot of pancreatic cancer research, where they have done a lot of whole genome sequencing, where they sequence every single genes in the cancer itself and lead to discovery projects and understanding. And this is where I really understood, as a clinician, I know how to look after patients, but to make a new therapy for that is good for my patient requires the scientists in the lab working out the mechanisms. But then I don't really understand the ins and out of a scientific experiment. These days, because we're working with DNA and genomic changes, and our genes, there are 19,000 genes, they all change according to one drug effect. How does that all integrate? We're not a single cell organism where you do one thing and one thing changes. We are a complex organism, and so to understand that, we need the big data, so bioinformatician crunching the numbers and computationally, and to understand that. And as an oncologist, I didn't understand how do you actually crunch the data, how do you leverage the gene changes that you see and help you understand what you see in the lab, and help me understand the direction I should go in my investigative care for my patient at the bedside. And so it's really these three pillars that need to come together. And that was a changing point. Working in that environment really changed my view, is to say, I need to be across all three, be able to talk the language and break down those barriers so that we could be effective. And that's really brought me back into Melbourne, where in Peter MacCallum Cancer Centre in Melbourne, we're able to have the scientist, the clinician, the bioinformatician all housed in one institute. And that really breaks down the institutional barrier to collaborate and really make a difference in terms of developing new therapy for our cancer patients of tomorrow. The research areas of interest is RNA biology. What is that? That we have our DNA, that's a blueprint of our body, but we got to turn that blueprint into building block, which are proteins. The way the message gets out is this RNA intermediate steps. And to me, I call them, they are words in a sentence. Words on their own doesn't make much sense, but string together in a structural way, they make a lot of sense, and that's how we communicate. Cancer is clever. It hijacked this mechanism and made it more favorable for them to grow and more aggressively and takes over. But now we have ability of understanding how this language and interaction happens, and if we can remove the hijacking and hijack it for our own benefit and disrupt their hijacking, we may be able to save people from dying from pancreatic cancer. And the exciting bit is this is a new way of thinking about treating cancer, and it is through new ways we can find new discovery that may be the Achilles heels to pancreatic cancer that we haven't found today. I have been very fortunate. From the get-go, it was my PhD work allowed me to initiate a lot of these research proposals. It was through the NHMRC, which is a national medical research funding body, which supported two different groups, one from a melanoma group, one for a pancreatic group. But the melanoma group was really, they were experts in understanding molecular oncology and providing the wet lab support. The pancreatic cancer group out of University of Melbourne, they were the expert in the bioinformatics and analysis around pancreatic cancer research, and the leader of the group is drove a lot of the Australian Pancreatic Cancer Initiative, and to be able to bring those two groups together and allow me to really facilitate a truly translational space, working on three pillars, with me being the clinician, having the wet lab and the dry lab collaborating and working on this new project. And through my PhD, I was also supported by PanCare, philanthropic support, having extra funding to fund some of the sequencing study that are very expensive to do, and having these extra funding and allow me to look deeper into the studies that I wanted to look at. Then that was further supported now in the sort of postdoc era is through the funding of Dare to Hope, and in collaboration with PanCare, they again able to fund the continuation of that research spark that I started from my PhD, and be able to run with the story and see it to a fruition. Whereas a lot of project out of PhD you might stop because that's where the funding stopped. But through philanthropic support, I'm able to continue the passion and really drive at the question whether this targeting RNA could be a way of targeting pancreatic cancer. I think the passion for research comes from the day-to-day clinical practice. I see my patient, I see what I can do with the limited drugs that I have. Chemotherapy only gets so far for pancreatic cancer patients, and I really want to go above and beyond and do that next level for them. But being involved in research, I'm really at the forefront and understanding what molecular mechanism might be driving a cancer. And this is the forefront that we sort of being told about, the precision medicine, where we have so-called targeted therapy for pancreatic cancer. But as I know, this is only a very small part for pancreatic cancer. I want to broaden that horizon. So this is where I look to drive lab research to look at different fundamental mechanisms that we can target, and that allows me to do better for my patients, so that when I get back into the clinic, I have ability to tell my patient, yes, there is something promising else I'm working on. It may not be the drug that you'll have tomorrow, but this is a step in the right direction. At the same time, I can sit in front of my patient and I go, because I breathe and read about all things about pancreatic cancer, I can confidently say I am the expert in pancreatic cancer, and these are the trials. And yes, you can bring me the trials that you read about, but I can put that into context for you because I know the data like the back of my hand, and I can place that into context. And that brings value for me as a clinician because I can make the undigestible into a digestible form and put out a clear message so you have confidence that you have a plan going forward, so you are empowered to decide what you want to do with your future. I think outside of work, because I do research and takes up so much more time, my private practice and clinical work takes up the other half where I'm caring for my patients. It leaves me very little time to enjoy. And so what little time I have, I go back to my kids. They are my pride and joy. A frustrating work day at home, and yet you come home to two smiling faces, and that really helps me to overcome sometimes the disappointment with research when the research outcome wasn't what you were hoping for, but you are able to sustain yourself to keep going. And that, oncology has taught me what shines through the tough times is your family. And family really brings that out. And outside of that, I really enjoy cooking with my wife, traveling and making different experiences. And so that my kids see a different part of the world, and that are so privileged to be able to move and be in a different place and see how they grow as their eyes are open to new events around the world. And so that we really enjoy that as a family.